Hello students, good evening to all of you all. Assalamu alaikum. Welcome to the online session. Can you all hear me clearly and can you all see the screen clearly? Shared screen clearly.
Yes, sir.
Okay, good.
Yes, sir.
Yes, sir.
Thank you. So last week we were discussing about the full introduction about our biology, chemistry, physics, basic science introduction, and deeply we went into the biology introduction, right? Yes. Yeah, before starting, I'll just tell you, we have sent the tudes to many students. Some of them have received. In the middle, we got like short of tudes because for the physical classes, more than expected, the students came. So we gave them the tudes. About 20 students we were unable to send, but more than 35 students we have sent the tude. I think many of you all received the tude. The ones who didn't receive, don't worry. If you have filled that Google form, inshallah, within this week, I have printed it. Today only I got the new prints. I will be sending you all the tude. Okay. So we, from the institute, from the admin, now this is me. You all will send me the request. No, I will send it to the institute. Admin. The thing is they have the maths, commerce, ICT. So for them, the workload this week is very heavy, very heavy workload for them. Because our physical classes also more than expected, the students came, we were a little bit unable to, manage them. Classes got filled. We had to get more extra tables and chairs and all that problems happened. So they were having a hectic week. So please excuse us, the ones who didn't get the tude. Most probably within this week, we will be you all will be getting it. Okay, so don't worry. However, if you all have registered, you will be getting it. So from the admin, sometimes if you all have registered to science maths, they will send science and maths tudes together. Or if you all are only single subject, they might send science together. Or if my tudes are finished, only Rizlan sir's tude is there, then they will first send the maths tude, and after my tude come, they will send it. So it goes to different parts of Sri Lanka, and finally, you will receive it. Okay, finally you will receive it. So don't worry. We will be delivering it to your home, and there is no any extra charges or hidden charges for the tude or the postal. Okay, so first month, all of the students who are registering to the WhatsApp classes or through the WhatsApp group who are registering through the Google form will get it. From the next month onwards, that means our second tude onwards, the ones who purchase the classes, as soon as you purchase, your name will be printed, then like that they will send. So from the upcoming tudes, it's very easy. First tude is free, no? So we have a lot of workload to do. So please understand our situation also. We will be sending it. Okay, so that is the thing about the tude. So don't worry, I will be mentioning the page number of the tude. Actually, the ones who were not there last week, this is the tude which you all are receiving. The grade 9s, they will be receiving this tude. The grade 11s, most of them already they have received this tude. This is our grade 11 tude. This is our grade 9 tude. Grade 10 tude. And this is the grade 9 tude. Okay, and there is another set of tudes. Next, most probably by March, we will be sending. That is the second part of first term. All that also you all will be receiving. The ones who are continuing the classes will be receiving it. So I hope you all went to school and showed your colors. Some have shown the tudes to the friends and they also wanted the tudes. I have put them in the request list and they will also be getting it. So good. And some of the parents contacted me and told that the students are normally for the online class, they are just going here and there and they are not focusing. But in my classes, I got to know that the students, some parents said they thought online is not success. Students won't listen, but Alhamdulillah, in my classes, they were focusing, they were drawing low, they were writing low. The parents also was shocked. Okay, so good. So I hope with this presentation things are being made more easy and more interesting. So like this we'll continue. And I have seen tremendous results from the online side. So okay, online students are the ones who are scoring the highest percentages of A, more than my physical class student. I scold them sometimes, some physical class student for fun, I just hit them also. Even that, they are not studying. The online students are doing better. I think that is because of the recordings. They are watching it again and again and getting everything into their central processing unit, their brain. Hari, so last week we were discussing about the carbohydrate. We discussed about monosaccharides, disaccharides, and polysaccharides. I think we were able to complete only monosaccharide, no? Yeah, we discussed this chart, if I'm not wrong. Yeah, the grade 11 students, there are some grade 11 students here, no? Grade 11 students, you will receive the free tute for grade 11. If you want the grade 10 tute also, you all can purchase the grade 10 class. The last two classes are there. Once you purchase, you will get it. If you all are continuing the grade 10 classes, you all can purchase the last two classes. And also from next month onwards, for the grade 11 students, after purchasing your grade 11, you will be paying the full fees. When purchasing grade 10, the ones who want to come to grade 10, you don't need to pay the full fees for grade 10. You can pay half and join. That is for the grade 11 students who are joining grade 10 also. You all can pay half the fee and you can join. No problem with that. So grade 11 students will get the grade 11 tute freely. Grade 10 students will get the grade 10 tute freely for this month. And also the grade 11 students who purchase the grade 10 classes, remaining two classes, third week, fourth week, they also will be getting the grade 10 tute. Because we are limited amount of tutes only there. I thought of sending for all the grade 11 students one free grade 10 tute, but not possible. Okay. Hari, then let's go into the discussion. Did we discussed about this maltose, sucrose, starch, maltose, sucrose, lactose?
No,
Just, yeah. Can someone tell me what is the last topic we discussed? Page number in the tute, if you can remember. Page number 5 completed, right? This part? Yeah, this is in page number 6, sorry. Yeah, monosaccharide table done. Now we'll go to disaccharide. Disaccharide, as you all know, it is made out of two monosaccharides. Glucose plus glucose will form maltose. Glucose plus fructose will form sucrose. Glucose plus galactose will form lactose. The common general formula of disaccharide is C12H22O11. Last week I told you all how this 22 and 11 is coming. You might think it should be C12H24O12, but why is this H22O11 coming? Because one water molecule is getting removed. All that we have discussed. And also the ones who didn't come last week, you all can watch the recording. It's available in the group. Again, I will upload it. Then you all can watch it. So the ones who have the tute, you all can draw this part. Glucose plus glucose equals maltose. Glucose plus fructose equals sucrose. Glucose plus galactose equals lactose. That part. And the places where it is present. Okay. Present in germinating seeds, malt, grain, and maize. Present in white sugar, brown sugar, sugarcane, especially the sugarcane juice, beetroot juice, some fruits, and the phloem sap. You all know, small grades you all studied, xylem transports water. Phloem transports food. What is the food that is being transported? Sucrose. Phloem transports sucrose. And under lactose, you all can put present in dairy products such as milk, yogurt, curd, cheese. All those things contain lactose. Page number 6 in the tube. I have given a space. You all can draw this and write the places in which those are present. The ones who don't have the tute, just roughly write these things. Once you get the tute, you can clearly copy the needed things. Okay, done with that? Or take a screenshot. You all can copy it later. Yes, under maltose, in the tute, I have given another extra additional point. An intermediate product of hydrolysis of starch. Last week we saw what is this hydrolysis of starch. What happens during hydrolysis of starch? Starch will initially break down to what? Maltose, and then finally it can break down to glucose. The final product of starch hydrolysis, the meaning of hydrolysis, last week we discussed breakdown, cadenova, breakdown. Final product of hydrolysis of starch is glucose. What is the intermediate, middle product? Maltose. So how can we get maltose? When we break down starch, initially we will get maltose. Later only we will get glucose. Okay, so that is known as the intermediate product of hydrolysis of starch. That is the additional point given in the tube. Then under sucrose, I have mentioned transported within the plant through the phloem tissue. Transport substances in the plant. So if I say there is xylem tube, this is the xylem tube. Like that, we have another tube which is known as the phloem tube. So xylem is transporting water. That is not related here. What is related here? The sucrose particles will be transported in the phloem. It can go upwards or it can go downwards according to the necessity. So what is the substance which is transported inside the phloem? Sucrose. Sucrose particles are transported up and down. The ones who have the tube, can you draw a rough diagram, small simple diagram like a tube, phloem tube, and sucrose is traveling inside that. Phloem tube and sucrose is traveling inside the phloem tube. Then lactose. Maltose, sucrose, lactose. We are talking about the disaccharides. Lactose, the only sugar which is not present in plants. So not present in plants. Plants do not have lactose because lactose is present in dairy products. Where can we get dairy products? Milk. Milk of the mammals. So cow, the female cow, once it gives the baby, it will start producing milk. Cow's milk, goat's milk. Like that, a human female, once she give birth to the baby, mother will be breastfeeding the baby, no? So human කියන්නේ female breast milk, that is also having lactose. So mostly related to animals. And also they have given the lactose percentage. If you take the cow's milk, the percentage of lactose is only 4 to 6%. If you take the human milk, that is about 6 to 7%. That's why they say, do you know that the doctors say for 2 years, the baby should be given mother's milk, not the cow's milk, not the box milk, because mother's milk has more lactose, more nutrient, more medicine for the child. Mother's milk is a medicine for the child. So human milk, that is, we are talking about the mother's milk, that is having more lactose percentage, more nutrients, more better for the child. So 6 months, baby has to be only fed with mother's milk. You can't give any other food. After 6 months, you can give other foods plus mother's milk. Now, can you understand from the percentage why the mother's milk is important for the baby? Yes,
Yes,
highlight
sir.
this percentage. Already in the tube it is given. Take a highlighter and highlight this percentage. Cow's milk 4 to 6%. Human milk is 6 to 7%. Human milk kyanne the breast milk of the mother. Any doubts or any problems? With that, one very, very, very important point I forgot to mention. If you look at lactose, it is not sweet. All the other, generally we say disaccharides are sweet, soluble, crystallizable, but the lactose example, it is not sweet. If you take normal cow's fresh milk and drink, there is no sweet taste, no? After adding the sugar only, you get the taste. Nathang, it's not sweet taste. It's just milk. Okay, milk is not sweet. So lactose is not sweet. Even galactose, we spoke about that, no? Even galactose is not sweet. But generally, when saying disaccharide, normally we say it's sweet kila. But example wise, when you're checking, sucrose is sweet, maltose is sweet, but lactose is not sweet. Are you clear? That is a very important point. Huh? It's not sweet. Yes, now let's go to polysaccharides. There are 3 examples for polysaccharide: starch, cellulose, and glycogen. Page number 7 in the tube. Examples, write it down. Grains, yams, jackfruit, and breadfruit, they are having starch. And all the products from that, for example, even bread, even bun, the wheat flour, that has starch. Cellulose is present in the cell wall and plant fiber. Glycogen is present in the animal liver and in muscles. First, we'll write the examples and or the locations, and then we'll discuss about it. Starch, it is stored in the plant, so that is known as the plant storage. I will show a small chart related to that. You all tell me, as soon as photosynthesis happens, what is the immediate product of photosynthesis? Last week we discussed a little bit about this. What is the immediate product of photosynthesis? As soon as photosynthesis happens, what compound will be formed in the plants? Anyone?
Glucose.
Yes, glucose will be produced. Okay, this glucose cannot be kept for a long time in the plant. So glucose will be converted to starch and it will be stored in the leaf. Then if the other parts of the plant need glucose, glucose has to be transported. So before transporting, glucose or starch has to be transported. You can't directly transport it as glucose or starch. What should you convert it to? Previous slide we studied that. It should be converted to sucrose. And through the phloem, it will be transported. And then it will go to the needed part. For example, let's say root. And whatever needed to be used up, it will be used up. Whatever excessive, what will happen? It will again convert to starch and get stored. It may be stored in the roots or stem or any other parts. So whenever carbohydrate is being stored, in which format is it being stored? Storage in leaf, storage in the roots or stem or other parts. In which format? Starch format. So that's why we say starch is the storage carbohydrate in plants, not in animals. In plants, storage is done by starch. Soon after photosynthesis, glucose is coming. Glucose is temporarily stored in the leaf. Okay, it is temporarily stored. After that, it is converted to sucrose and transported. It will go to different places, the roots, the leaves, the fruits. Whatever extra is there, again will be converted to starch and stored in the other parts. So whenever storing carbohydrate, it will be stored as starch in the plants. So that's why we are saying storage product or storage carbohydrate is starch. Yes. Page number 7, under starch, can you all just slightly write this? There is a small space. In that space, you can just write glucose, then starch, sucrose, and starch. Starch is the storage product. Store kila danna. Yeah, I'll show the previous slide after this. Because if I go back again, all this will get deleted. That's it. If you have any doubts with this, please ask. Clear, no? Let's go back to the previous slide. Take a screenshot if you all have not written it. So Balanna, stored in the plants. Now you know the full story of this. First glucose. Glucose will be converted to starch and stored in the leaf. After that it will be transported as sucrose. And after that when it goes to the needed places, again it will become starch. So starch is the storage product of plants. Clear about that? Now let's go to cellulose. When you look at cellulose, cellulose is present in cell wall of plants. It's a very important point. Cell wall of plant is made out of cellulose. The other place where cellulose is there is fiber. This fiber is not digested by the human digestive system. Therefore, it helps to avoid constipation. Small grades you all have studied, no? To prevent constipation, you have to do two things. Drink more water and eat what rich food?
Fiber.
Fiber rich food, no? So fiber rich foods are present in plants and all those things. So that will avoid constipation. What is constipation? You're nicely eating, but not going to the toilet. Not passing fecal matter. Very hard to pass fecal matter. Because sometimes the fecal matter has got pasted to the walls of the intestine. So this fiber will clean everything and come out. So fiber helps to prevent what? Constipation. Fiber helps to prevent constipation. So cellulose, it is present in cell wall, it is present in the fiber, and the fiber helps to prevent constipation. Constipation is opposite of loose motion, no? What is loose motion? Even if you eat little, going, output is going. But
Loose motion.
constipation, okay, constipation means not going. So to prevent constipation, they are telling us to eat a lot of fiber-rich food. Next one, glycogen. Okay, there is a important thing about glycogen. Yes. Now, when we eat rice, what is rice? Starch. Chemical component of rice is starch. Starch will break down to one product. I want you all to name it. Starch will immediately break down to one product. When we put in the mouth and chew that and eat it,
Glucose.
swallow it, it will first break down to maltose and then break down to glucose. Okay. Earlier also I showed you, no? Intermediate product of hydrolysis here. Starch, maltose, and then glucose. Now what will happen? This glucose will be absorbed into our blood. Now in our blood, what is there? Glucose. Needed glucose will be used to produce energy. Glucose only produces energy, no? So after producing the energy, if glucose is remaining, what to do to that glucose? It has to be stored. Can it be converted to starch and stored in the human? No, can't. So glucose is converted to another product. Remember, glycogen. And this is stored in animal. So now you all know animal storage is glycogen, plant storage is starch.
The starch means carbohydrate foods.
Yeah, carbohydrate food. Starch means earlier we studied grains, yams. Now here carbohydrate is a main, like a broad topic. Now we have studied 9 carbohydrates. Glucose, fructose, galactose, maltose, lactose, sucrose, starch, cellulose, glycogen. 9 are there. So starch is normally present in rice, grains, yams, even the bread, bun, potatoes. Those are also having starch. So the excessive glucose in our blood will be converted to glycogen and stored in the animal where? 2 places. Liver, muscles. And also once the muscles are filled, it will be stored under our skin. That's why people become fat. When they eat too much of glucose, we become fat, no? Because it is getting converted to glycogen and stored under the skin. Okay, now I want you all to answer, especially the grade 10s, not the grade 11 students. Huda, inside our body, there are hormones now. There is one special hormone which can convert glucose to glycogen. Glucose to glycogen. That is naturally produced. For some people, naturally it's not getting produced due to some damage, internal damage. Then they have an injection. They keep it in the fridge. Soon after eating or sometimes before eating, They take that injection and put it in their stomach. Your grandfathers, grandmothers, or aunties, mothers, fathers, they might be doing this. What is that injection known as? That is the name of the hormone.
Insulin.
In-
Yes, insulin.
in-
Okay, so naturally, thank God, insulin hormone is there in our body. Therefore, we don't need to put the injection. If our insulin hormone is lacking only, they have to put that injection. Why do we need insulin for our body? Excessive glucose is converted to glycogen. Or else if too much glucose is there in our body, what is that disease known as? Too much glucose in the blood is
Diabetes.
diabetes. We say sugar. We say seeni. Seeni වැඩි වෙලා seeni ව්‍යාධි. It's not seeni ව්‍යාධි. Its proper name is diabetes. Are you all clear about it? So under glycogen, just write this part. Glucose getting converted to glycogen with the help of insulin. Just write this part only. So now you all are clear. Animal storage glycogen, plant storage is starch. If you have any doubts, please ask.
Sir, glycogen is important to our body.
Yeah, it is storing all the starch and extra glucose and keeping. Later we can use it. Now if you buy bread, you eat the bread. Balanced bread, you put in the fridge or store it somewhere and keep. No, later, tomorrow morning we can use Kiela. Like that, soon after we eat, there is more glucose. Glucose will be stored as glycogen. Later, when we are becoming tired, when we are doing work, glycogen will be used to produce energy. Okay.
Okay, sir.
Yes. Yes, next we have to go to the test to identify carbohydrate. How can we, some of the carbohydrate can be identified in the laboratory by doing some tests. I'll explain about that tests. In schools, you all might do those tests. Yes. Shall we go to the first test? The ones who want to take a screenshot of this, you all can take. Okay. Iodine starch test, or they call it iodine test. Okay, when starch or starchy solution or starch food comes in contact with iodine solution, a purplish blue color appears. Actually, Puta, here you can see iodine. The color of iodine is yellow or brown. Sometimes it's like this. There is a Mara argument between school teachers. In some schools, listen to this carefully. Yeah? In some schools, they have pure iodine, which is dark brown. In some schools, they have mixed the iodine with water. It is light. It's like yellow color. So one school teacher is saying in our school, iodine is brown color. So we have to put brown. The name should be brown color. Another teacher is saying no, yellow. Both are same. In one water is added, in the one water is not added. So we'll keep in mind, if they ask you what is the color of iodine, we have to be like little bit tricky, cunning. Symbol of iodine is I2. Color we will say yellowish brown. Then both are covered. No, some teachers are saying yellow color, some teachers are saying brown color. When we say yellowish brown, both are correct. Then when these yellowish brown things touch a starch, or for example, let's say we have a starch solution. we have taken some wheat flour and mix it with water. It is starch solution. Starch solution. Wheat flour plus water. Now to this we are adding few drops of iodine. What will happen? The color will change to a dark purple color. Okay, there is a problem. There is a problem. If the teachers are using pure iodine, it will give a dark blue color, very dark blue color. If the teachers are using that water mixed iodine, then it will give a purple color. So again, there is a problem. Some teachers are saying we are getting dark blue. Some teachers are saying we are getting purple. So to solve that, what are we saying?
Purplish
Dark
blue.
purplish blue. We use the word dark purplish blue. So you can identify starch when you put little bit of iodine, yellowish brown iodine will convert to dark purplish blue. Are you clear how to identify starch? Now here there is already experiment they have done. They have taken a potato and added few drops of this. Can you see the color? It's like a dark blue color, no? If you properly see, if it's a real photo, if you go close and see, you can see like a purple shade is there. Okay. So that's why we are saying dark purplish blue. All the needed things are there now in that. Can you all draw this diagram in that space? Page number 7. Page number 7. Next to this writing, there is a small space. Just draw this test tube and yellowish brown converted to dark purplish blue. When you add that yellowish brown thing, it will turn dark purplish blue. Okay, this topic I told you first day itself, one of the longest topic and most boring lesson. Okay, I'm trying to put all the pictures and explain and make it more and more interesting, but actually it is a boring lesson. In schools, some students are sleeping in this lesson. One is they can't understand and one is it's boring. But I hope with these pictures and all, you all are active, you all are not sleeping. You all are listening to it, drawing the needed diagram. I hope you all are doing that.
Sir, what is iodine?
That's a liquid solution. It's a chemical solution. Like water is H2O, iodine is I2. The chemical symbol is I2. It's available in the laboratory. You can put that to test starch. Are you all done with the diagram? We can go to the next one. So one test done. Like this, there are 10 tests in this lesson only. Iodine test, Benedict test, Burette test, Sudan 3 test, nitrogen test, water test, carbon test, copper sulfate test, cobalt chloride test, like that. 10 tests are there we have to study. That's why this lesson is more boring and long lesson. Actually, they shouldn't have given first lesson itself this lesson, because some students get fed up. Okay, they come more motivated. I have to study science from grade 9. When they get passed to grade 10, they are coming with full motivation. When they see this lesson, when they try to study this lesson, and when they can't understand, all their motivation falls down. So don't worry, other lessons are not like this. This is the boring lesson, new names, lot of experiments. Other lessons are more and more interesting. Okay. So don't lose your motivation. Don't lose your hope. You can go for a good results. Just because this one lesson is hard, don't think all the grade 10 lessons are very, very hard. Okay. Only hard thing in this lesson is new names. All are new to you all. Okay. Shall we move on?
Okay,
The ones
sir.
who have not written, take a screenshot because some
Okay, sir.
students slowly like a baba only they are drawing. So you take a photo. Later you all fill the tube. Okay, let's move to the next one. Here nice picture is there. They have added the iodine and you can see a dark blue color thing, no? In a
Yeah.
piece of bread. Bread is starch. Hurry, next one, Benedict test for monosaccharides and some disaccharides. When few drops of Benedict solution is added to a test tube containing glucose solution and heated over a water bath, the following color change appear. I will explain this. Okay. Now, we have to take a monosaccharide or some disaccharide. Who are these some? Alright. Glucose, fructose, galactose, all the monosaccharides will show color change. Maltose and lactose also will show color change, not sucrose. Keep that in mind. All these five fellows will show color change with Benedict's solution. Benedict's solution is a blue color solution. So what should you do? First we have to take a test tube,
Sir,
fill it
what
with
is mean by
glucose.
Benedict solution?
Sorry, Buddha?
What is mean by Benedict solution?
It's also a chemical. It's available in the lab. Okay. So first we have to take glucose solution. How to take glucose solution? When running for the sports meets and all, you have glucose packet, no? You take that packet, take 1 teaspoon, put it into water and mix. That is glucose solution. Then in your school lab, there is a bottle called Benedict solution. That one already in the chemistry factory they have produced. Add few drops of that. Okay, to that dropper, you take few drops of Benedict solution and add it. Now this will be blue in color. Okay. Now you have to heat it. If you directly heat it, quickly the color change will happen. We can't see very fast. So to slowly heat it, we use one method. That method is known as double boil method. In cookery classes, have you seen how they melt chocolate? They put
Yes.
chocolate pieces. They don't directly heat it. They keep it in the water and they heat the water. So this is known as what method? Double boil method. This is water. In the laboratory, you will keep a tripod. And what is the heating equipment present in your school laboratory? What burner?
Bunsen burner.
Bunsen burner.
Bunsen. Bunsen burner. And this tool is the tripod. It's a metal one. And you all can keep, this is a, I'll make the beaker small. A small beaker like this. And this is a test tube. Or a small tube is known as boiling tube. Test tube, small one is known as boiling tube. You can use test tube or boiling tube. You have added Benedict solution. Few drops of Benedict solution. Now when you heat it, now it's already blue color now. From blue color, it will become bluish green and turn to green color. Then
Sir,
from
is it
green color, it will become greenish yellow and finally it will become yellow color. From yellow, it will become orange color. And then from orange, it will become a brick red color. Red color, we call it brick. Gadol, you all know brick. Brick red.
Sengal?
Rasi, brick, Senkal, brick red precipitate. So from blue, it is turned into green. From green, it is turned into yellow. From yellow, it is turned into orange. And from orange, it is turned into red. Finally, it will be red in color. Easily you can remember this. Can you all remember the short form for the rainbow colors? Small grades you would have studied Vib G Yor.
Geo.
Vib G Yor. Okay. So start from blue. Forget about this V and I. Blue to green, green to yellow, yellow to orange, orange to red. Finally, brick red precipitate. Finally, it will come to the bottom. Precipitate means like it get deposited at the bottom. Yes. Can you all draw this picture, this small setup in page number 7 next to the Benedict test? And then below that there is a space. Write this color change. Now, because I don't have space, I have brought it down. You all can write it in one full line if you have space. Blue, green, yellow, orange, and brick red precipitate. Precipitate short form is PPT. You can put brick red PPT. Blue, green, yellow, orange, brick red PPT. Are you all clear? If you have any doubts, please ask. After doing all the 10 experiments, I will show the video. Okay, the YouTube videos are there related to this experiment. I will show that. But you don't need to watch the video to understand. When I explain it one by one like a baba like this, you can easily understand it. No? Take glucose solution, add Benedict solution, mix it and slowly heat it. To slowly heat it, you need a water bath. Okay, a beaker filled with water. That is known as double boil method. Then only slowly it will get heated. Then you will be able to see the color change. Blue to green to yellow to orange to brick red precipitate. I hope that is clear. Maximum I have explained. If you have any further doubts, please ask.
Sir, what is precipitate?
Precipitate is like finally it will get deposited at the bottom. Now, if you take a juice and keep for some time, heavy things will come down. No, like that it will get at the finally you will see heavy things down, red color heavy things at the bottom. Any other doubts you all can ask? Please ask the doubts, because in schools I got to know some students are getting scolded for asking doubts. Don't ask unwanted doubts. What is this? I taught you know again and again. What are you asking this? Go home and study the lesson and come like that. No, I got to know some complaints. Don't worry here. That is not there. You ask the needed doubt. I will explain the needed things. Yes, quickly take it down. Only thing is,
So,
one thing,
so,
make your drawings fast. Yes, Buddha.
so in the exam, how will they ask this Benedict solution?
They might ask, what is the color change? Okay, or else they might give you something like this. How there is a test tube with glucose. What is the test done to identify it? Benedict's test. What is the color change of that test? Blue to green to yellow to orange to red to brick red precipitate. Like that they might ask. Or they might ask, why do you boil it in double boil method? Okay. Here, to heat the solution slowly. Like that, different patterns, they might ask the questions. So that's why we are learning the theory clearly. Whatever type of question they're asking, we are able to answer it. If you know the theory properly only, we can answer. That's why in bio, if you know, you know. If you don't know, you don't know. How are we going to answer? That's why I'm like a baby, like a parrot, I'm explaining to you these bio parts.
Sir, are there keywords to write when we write the definitions in exam?
No, some places there are some keywords, but here nothing like that. Those are coming for definition. For example, they might ask you what is growth. Later you will study that. Growth is the irreversible increase in the dry size of mass. That irreversible word is important, but here nothing like that. Only the color change is important.
Sir, will we get some activities like this in the exam?
Yeah, sometimes they might give. They will say there were 2 test tubes which were unidentified. So to test tube A, you added iodine, it showed a blue color change. To test tube B, you added Benedict's and heated, it showed color change. What is in test tube A? What is in test tube B? Like that they can ask.
Sir, do we have to draw it?
Drawing part might not be there. Now here in the theory you have to draw and show. But in exam, they won't tell you to draw. Now, these are simple experiment. We have other many important experiment where they tell you to draw and label, or they give the picture, they tell you to label. These are, now no, you all are new, no. You might think, oh, this is a very important experiment. Ayyo, this is useless experiment. We have very, important ones. These ones, they might ask only questions related to this.
So is this important for all?
Yeah. Whatever we are studying from grade 10 first day onwards important for O-level. I can show you there are many O-level questions related to these tests. That's why I am having the past paper classes. Soon after Ramazan, we will start our paper classes, okay? That is very important. In that, all the past papers from 2016, I have selected it separately. Okay, later you all can check it and see. 2021 O-level paper. Second question they have asked about the amylase test. That test is they have asked. Then 2024 last year paper MCQ question number 21. I'll read the question because I can't show it in this. I'll just read it, just see. No, still you have not studied that. That is related to Benedict test. Okay. No, after doing the next experiment only you will understand that. It's related to the Benedict test. Okay, so like that in different places they are asking these questions. 2016 there is a question related to carbohydrate, proteins and lipids. Separately they have given what is the enzymes, what are the digestive products and all those things. So like that in past papers, many places they ask. So whatever you're studying from grade 10 first day onwards until the grade 10 last day is needed for your level. We don't know which question they will ask, so we should know everything. Hurry, shall we move on? Now we are going to the, if there is any doubts, you all can ask. Quickly ask, we'll go to the next part. No. If there is anything, just send me a chat box message. Okay. They have shown after the experiment. Can you see? Brick red precipitate. Now we know that this. Test for sucrose. Because the sucrose doesn't show color change with Benedict. So what should you do? You have to do this fellow is a little stubborn fellow. You have to do something to him. We'll see. Take a sucrose solution and conduct the above Benedict test. If there is no color change, then take another similar solution, this sucrose solution, and add few drops of sulfuric acid. The formula of sulfuric acid is H2SO4 and heat it. Now add few drops of Benedict solution and do the Benedict test. The same color change will occur. I'll draw that and show. Is the paper class free? No, the paper class we will be starting in March, most probably soon after Ramadan. March month I will be giving it freely, and most probably April it will be a paid class. It will be just, I think, 1,000 rupees. Okay, every week we will be doing papers, past papers, unit papers, term test papers, provincial papers. Now we not enough theory. We have not covered enough theory to start the paper. By March we would have covered the this book. No, this book will be done by March. By the end of February we are trying to finish this book. March first week you will get the new tute. So March we will be completing the other part. By that time enough of theory is there. We can ask nice questions. Past papers we can do. Superb.
Sir, is paper class physical only?
No, paper class is online only. Even my physical class students will come to the paper classes online. Okay, I think it's on Friday. I will give the full details later. Rizlan sir is having his paper classes. Soon after his paper class, my paper class will start. Something like that. Or before my paper class, I think his paper. No, after his paper class, only my paper class. So Rizlan sir, you all know our maths sir, no? He's one of the famous person in Colombo, producing the highest amount of A's in Colombo. He's doing online classes. He's only the main person in Master Academy. So Master Online also you can purchase his classes. His theory classes are also happening. His paper classes are happening. So my paper classes will happen soon after his paper class finish. So Friday you all can keep for paper, maths paper, then science paper. But we are not starting it in January because February Ramazan timing, it will be clashing with the prayers and all those things. So in the middle of March, soon after the Ramazan festival, we will be starting our paper classes. At that time, you all also will have a lot of knowledge. You all also want to express that knowledge. Paper classes will be really nice.
Sir, do you
Okay.
taking OL papers?
Yeah, O-level papers. From O-level papers, whatever possible for you all to do, I will extract those questions and I will give you. That is one. The other part is unit papers are there. And provincial papers. Now, for example, Western Province, there are some papers. Southern Province, there are some papers. Some good schools are there. Like sometimes Jaffna Hindu College, Royal College, Vishaka College. From those and all, I will select and take questions. And we'll be trying those. Because grade 10 is full O-level practice, army training only. No chance to escape. If you all are thinking just to come to class and have fun and go, my class is not suitable. In our class, hard work, working, effort, because we are targeting results. Okay, I know there are some classes, even physical classes, students are going and just talking. In school also you all are talking. In classes also, if you all go and just talk with friends, no use, no. But there are some places in our classes and all, if you come to our physical classes, you can't talk. 1 or 2 words you can talk, but just jabbering and waiting can't happen. Can't use phones. Because in some tuition classes, last row fellows, they are slowly keeping the phone under the table and nicely playing games, watching cartoon or some movies. In our class, if we confiscate the phone, we give it to the admin, parents only have to come and take the phone. Second time they get caught, out of the institute. Okay, but online class I can't confiscate the phone because you all are doing the classes in the phone itself. So okay, but even in the online classes, okay, later on when we start papers, suddenly I will take a call and ask your parents, is he doing the papers properly? Send me the picture of the papers which he has done. Like that I will be suddenly asking. If you all have not completed anything, I'll give one chance. After that also, if you are not completing, bye. You can find for another very friendly, easy teacher who's not checking any work or anything. Okay, so maintain your tutes. Maybe by February, I might tell you, send the pictures of the tutes. And then I will check. Inshallah, we'll see. So, we take sucrose.
Are you teaching to schools?
Abda?
Are you teaching to the schools?
Yeah, I am teaching the school which I studied my O level. I am teaching there. I go part time and I teach and come because I can't go full time because I have a clinic. Okay, so 2 days I go, I teach in a school, and other things, mainly the institute. Okay.
Sir, can I know where's your clinic?
It's in Dehiwala, Hill Street, next to the Sarasavi bookshop. In Colombo, you all know Dehiwala is very famous for the zoo, no? Everyone has come to Colombo, Colombo, especially Dehiwala, because everyone has come to the zoo, no? To see through school or through some organization or with family trips. My house is also in Dehiwala. My clinic is also in Dehiwala.
Sir, near high level road.
High level road is, yeah, that's a little bit further away. It's in Nugegoda. Yeah, there is.
Near,
So if the
near the
solution
bridge.
gets cold, will the brick red color change back to the normal color? No, no. The color won't change back. There is a question, what is the similar solution for Benedict solution? Actually, for your O level, it's not needed, but there is another solution. Okay, two solutions you have to mix and put, but not there in your O level. Someone has asked, maybe you might have seen something in the internet, they would have said Benedict solution, and another nice name is there. I can't remember that name. Okay, that is not there for O level. In O level, we directly go with Benedict solution. Hari, Balanda. Now, when sucrose is directly put to the Benedict solution, failing solution. Yes, that's not there in our syllabus. Directly when you put the Benedicts and heat it, no color change for whom? Sucrose. So what should you do to sucrose? First you have to take sucrose. Then you have to add few drops of an acid. What acid? Sulfuric acid. The formula of sulfuric acid is H2SO4. Little by little, now itself get used to these formulas. H2SO4, sulfuric acid. And then you have to heat it. After adding the sulfuric acid, directly you can heat it. And after that, perform the Benedict's test. Already we know what is Benedict test, no? Earlier we discussed. Do that same test. Take it to a test tube, add the Benedict solution, keep it in a water bath and heat it. Then
Double
what will
boiling
happen? Then double boiling method. Yeah, that method. Then what will happen? Color change will be shown. What is the color change? Blue to green to yellow to orange to finally red. What type of red?
Brick.
Brick red. Short form is PPT. Brick red PPT. Precipitate. Are you all clear? Now we have done 3 tests. First one was starch test or iodine starch test. Second one, Benedict's test. Third one, sucrose test. That is also Benedict test. We first put the acid and then do the Benedict test for sucrose. Those are the tests available for carbohydrate. Yes, page number 7. Under that third topic, test for sucrose, just roughly draw this. That is in page number 7, last part. There is a question. So do we need to add sulfuric acid for the first tube or a separate one? Separate one. Because, that's why I said this is A, this is B. Because once you add the Benedicts and heat it, again you can't put the sulfuric and do. Separately, freshly you have to take. So this A is a fresh solution of sucrose. B is a fresh solution of sucrose.
So if the color change only, we know there is carbohydrates in it.
Yeah, here you can't say carbohydrate. We can say for this test, after putting sulfuric and then heating and doing the Benedict test, if color change is there, 100% we know it's sucrose. Without putting sulfuric acid, then directly when you put the Benedictine, if there is color change, we know it is either glucose, fructose, galactose, maltose, or lactose. But which one Kela, we don't know. For that, we have to do chemical analysis. But out of these 5, someone might be there. If you put iodine and it show color change, 100% it is sure it is starch. So these are the 3 simple tests. In A level, you will study more. Simple tests available for our O level syllabus. There is a question, will they ask how to perform the test? No, they will ask what are the chemicals added. If the Benedict solution color change is not shown, what is the additional thing we can do at the sulfuric acid? Like that, they might ask.
Sir, do we have natural disasters in grade 10?
No natural disasters, no ecosystems. All that charter lessons are done for until grade 9. When we put iodine to starch, will it react immediately or slowly? Normally, this iodine starch is very fast. Once you put, immediately that drop will turn blue color. Very quick. I told you, no, until grade 9, your science is a Acharu syllabus. Biology, chemistry, physics mixed and some unwanted lesson, lightning, that and this. But in grade 10 and 11, the lessons are superb lessons. Again, real scientific lessons. Whatever, important scientific ones now. Even natural disasters are important. But, that's not like into that real scientific thing. But these and all, you will understand in the next physics lesson, chemistry lesson, real science. Now, normally I say, grade 9, third term, I was telling each and every class, in grade 10, your real science starts. Yes, any other doubts? We'll go to the next part. Sir, in the step B, should we add Benedict solution as well? First, you have to add few drops of sulfuric acid and heat it. After that only you have to add Benedicts and do the Benedict test. When you are doing the Benedict test, however, you have to add the Benedict solution. No? After heating with sulfuric, then only you have to add Benedicts. You don't add Benedict's sulfuric and then heat it. First sulfuric, heat it, and now perform the Benedict's test freshly.
Sir,
Clear,
which
right?
part is grade 10 book? Which part
I
is
didn't hear
hard
you.
in
It depends. Now, this first lesson will be hard for many students because all new names and all those things. But we have chemistry part. Some people say chemistry is easy. Some say chemistry is hard. Some say physics is easy. Some say physics is hard. It depends from person to person. But if you learn it properly and clearly, like now you are getting all the clear explanation, no? If you learn it properly and clearly, nothing is going to be hard. You will, whatever hard questions they give, you will be able to answer. That's the thing. Okay, let's move on. Okay, functions of carbohydrate. Now, if you all want to take a screenshot of this, take it. Because once I erase this, it won't be there again.
Sir, you didn't add me in the group.
Putha, send me a WhatsApp message, will you?
Okay, sir.
Send me a WhatsApp message. I will add you. Most probably tomorrow morning, I will add all the other remaining students. Yes, now let's go to the functions of carbohydrate. There are 4 functions. Carbohydrate is the energy source. Carbohydrate is a storage component, structural component, and constituent of nucleic acid. Don't worry, I will explain one by one. Carbohydrate is the first point, energy source. The main source to obtain energy for the activities of all the organisms is carbohydrate. By hydrolyzing means, we have studied last week, breakdown of starch and other carbohydrate, glucose is obtained. This glucose can release energy after undergoing cellular respiration. Last week, the last topic, can you all remember, we studied separately about photosynthesis and cellular respiration. So again, I am going to tell you, due to cellular respiration, energy is released in our body. That energy only, now I am talking. My heart is beating. My lungs are working. I am raising the hand. I am doing like this. All this due to the energy from the glucose. So how does that happen? Okay, glucose. C6H12O6 plus, I'll take a new screen and I'll draw it. This is known as cellular respiration. What is cellular respiration? Oxidizing. Oxidizing means using glucose to produce energy. Where does this happen? Inside the cell, there is a small tiny organelle called, can you remember mitochondria? Last week we studied. There is a small organelle called mitochondria. Inside the mitochondria only this happens. So to produce energy, two raw materials are needed. One comes from the food, other comes from the air which we breathe in. From the food which we eat, glucose is there, C6H12O6. From the air which we breathe, what is the important component? Air has carbon dioxide, nitrogen gas, oxygen gas. Which gas is important for the living things? Oxygen.
Oxygen gas.
They will go inside the mitochondria and produce energy. While producing energy, carbon dioxide will be released, water will be released, and the main product is ATP energy. Can you all remember, I told you an easy method to balance this equation. What is the easy method?
Sir, what is meant by energy?
That is only the energy which is helping our body to do all the work. Okay, ATP, there is a meaning for A, T and P, adenosine triphosphate. For now, don't remember that, just remember ATP. In grade 11, we'll study the definition of ATP and all those things. Now, in the current wire, there is current electricity, no? Electric energy. From the light, we are getting light energy. Like that, what is the name of the energy which is making us move and talk and all those things? ATP energy. Now, this equation, to balance this, there is a short method. Okay, just remember my phone number. Not 778283. What are the last three numbers? 666. So, already this has 6, no? Don't need to put 6 there. In front of oxygen, in front of carbon dioxide, in front of water. When you put 666, it's a balanced chemical equation. Because earlier and all, we just write chemical equation, but now we have to balance it. So, there is a separate topic in chemistry for balancing equation. Here, I am giving a short method. Use that short method. Later, you will understand more deeply. What is the short method? Put 666 for cellular respiration equation and for photosynthesis. Both those reactions, the 666 method is okay. Yes. The ones who have the tute, page number 8, under energy source, under the first topic, you can write this equation. Already last week also we wrote, no problem, again we'll write. The more we write, the more it will go into our mind. Meanwhile, if you have any doubts, please send me a chat box message. Already there are some questions. No. Yeah, many have sent me the method to balance it. Yes, correct. So why you have put 666 to balance it? Later you will study the balancing method. For now, just put 666. That's enough. I'm giving a short method for you all. Okay. The ones who are not there in the group, here my phone number is there. So you save that number and send me your WhatsApp, through WhatsApp message, send me your name, grade, school, and from which area you are. Then I will add you to the group, and in the group I will send a Google form. You can fill that. Inshallah, we will be sending the tunes according to that. Okay, this is a simple thing. Listen to this carefully. Cellular respiration is the method of using glucose to produce energy. Now, we don't put the wire and charge our body, no. We need energy. So, our energy is from the food which we eat and from the oxygen which we breathe. So, the food which we eat becomes glucose and goes to the cells. The air which we breathe, from that the oxygen gas goes to the cells. Inside the cell, there is a small machine which is known as mitochondria. That mitochondria will use the oxygen, use the glucose, and produce energy. That energy only keeps us active and do all the work. While producing that energy, some other byproducts also will come. Those are the carbon dioxide and water. So for now, you just remember, glucose produces energy by joining with oxygen. What type of energy? ATP energy. While producing that energy, two waste materials are coming, carbon dioxide and water. Those waste materials will be later released. When we are breathing out, what is coming out? Carbon dioxide is coming out, water vapor is coming out. When we are sweating, water is coming out. Those are the waste water. Understood? So
Yes,
here
sir.
mainly I want you all to understand this equation, and now itself little by little practice writing the equation and balancing the equation. Will they ask to balance the equation? Yes, from second term onwards, all the equations should be balanced. That's why now itself I am training you all. Now I could have not like skip this part without giving the 666. I can just go. But now itself I am training you all to balance. Here I didn't give you like what to do to balance. I am just telling you memorize 666. It's not a big thing, no? If you know my number, my phone number itself, you will know 666. Used for photosynthesis and cellular respiration balancing. Done with that?
Yes, sir.
Yeah. The ones who have not done, take a screenshot. Let's go to the second use of carbohydrate. Harine? Okay. First function done. Produces energy. Second function, storage component or storage compound. You all know the answer for this. Answer again, you know the examples. What is the plant storage? What is the carbohydrate which is stored in the plants?
Starch.
Starch.
Animal storage?
Glycogen.
Glycogen.
Glycogen. Very good. So those are the examples for storage component of carbohydrate. That is a function. Carbohydrates can store things. It can act as a storage thing. Then structural component. Here, cell wall is a structure. It's a part of a cell. Who makes that cell wall? Which building block makes that cell wall? You all know the answer.
Cellulose.
Very good. Cellulose. Cell wall is made out of cellulose. That is a good example to show that carbohydrate can act as a structural component. Then next one, constituent. Constituent means part of. Nucleic acid. Now you all don't know what is nucleic acid. You might know that there are two types of nucleic acid, RNA, DNA. Can you all remember when studying the microorganisms in grade 9, virus is made out of two parts. Protein part, we call it the capsid, and either RNA or DNA. We studied like that, no? Okay. So these nucleic acids are RNA, DNA. The nucleic acid is made out of three parts. Out of that, one part is carbohydrate. If the carbohydrate is not there, carbohydrate is only holding the other two parts. If the carbohydrate is not there, other two parts will fall down. Okay, later there is a topic now. For example, if you turn to page number 15 in your tute, the full page, two pages are related to, one full page is related to nucleic acid. At that time, you will understand what is nucleic acid. Now, don't worry. The nucleic acid, to make the nucleic acid, you need carbohydrate, or else nucleic acid will fall down.
I didn't throw at you. So let us place.
So, just draw this structure. Now, under storage component, write plant storage starch, animal storage glycogen. Under structural component, write cell wall, cellulose. Under constituent of nucleic acid, you have to draw this structure. One nucleic acid is like this. This part is known as phosphate group. This part is known as nitrogenous base. All are new names, huh? Who's holding this phosphate group and nitrogenous base? This fellow in the middle, he is known as pentose sugar. He is a carbohydrate. Phosphate group, pentose sugar, nitrogenous base. The P and N is held by this S. This S is a carbohydrate. Pentose sugar is a carbohydrate. Just draw the structure and keep. Even if you don't understand, don't worry. Once you start our nucleic acid part, you will understand it better. That is coming a little bit later. For now, if you know the structure, that is enough for me. Phosphate group, pentose sugar, nitrogenous base. Out of that one part, the pentose sugar is what? Carbohydrate. If you all are done writing, you all tell me, then we can go to our new topic, protein. Carbohydrate done. Yes. Okay, then there is a problem. Problem can how to join the classes. Okay, now this is January. No, we have 4 weeks. First week, second week, third week, fourth week. So Putha, we decided from the institute to give the first week and second week free. So first week we completed it last week. The recording is there in the group. You all can watch it. The ones who missed it. Second week also free. If your friends have missed it, I will send the recording in another 2-3 days. Then you can share it to them also. How to join third week and fourth week? You can purchase the classes. Totally, the full month fee is 2,200. No, is it that exact fees mentioned in that leaflet? You all are grade 10. No, sorry, grade 10. Grade 11 is 2,200. Grade 10 it's 2,000. Grade 9 it is 1,800. Full month it is 2,000. So this month you don't need to pay this 2,000 because the first 2 classes are free. You can pay 1,000 rupees. And you have to upload the receipt to our website. Because now first week, second week, I gave the link in the group. Third week, fourth week, link won't come in the group. We have an online website. The ones who used to come to our grade 9, you all know. masteronline.lk, and also the ones who are already going to Rizlansa, you all know about this website. Okay. So when you go to Master Online website, first they will ask, first you have to register. If you are a new student, you have to register. During registration, they will ask your name, grade, school, address. Give the proper address. So that address only our future tutors will be coming. Okay, this is how to join the online method. And then they will ask you a phone number. Give a proper phone number, because if any emergency, any class related messages, if your payment is approved, all those messages will come to your phone number. And also give an email address, so that if you need to share the Zoom the recording, they will share to your email. Like that, we need the email address also. So give all the proper details and first register. During registration, if there is any problem or if there is any issue, you all can send me a WhatsApp message. I will tell the admin, because admin only checks all those things. They will look after it. Okay. After registering, when you enter the website, they will show you local syllabus, Cambridge syllabus, Edexcel. First, you all select local. Then in that, they will show you grade. Grade 9, grade 10, grade 11, grade 11.5. Select grade 10. And then it will show all the subjects. Maths, science, ICT, commerce. In that, you can select the science one. And add the classes to the that add to cart is there. No, you can purchase the third week and fourth week. And then directly you can do a online payment using the card, or else you can transfer the money to the given account in the website. They will give you an account, transfer it and upload the receipt to the website. Because when you transfer, we don't know, no, you have to upload the receipt. If you don't have that online banking or transfer method, you can deposit. You can deposit to our bank account, institute's bank account, and you will get a receipt now from the bank. Upload that photo, and the admin will check everything and approve it. So don't do it in the last moment. Let's say class is next Monday, 7 p.m. Don't do it at 6:30 or Monday 5. Try to do it a little bit early, then they will check it and approve all the needed things. For now, I'll just show you the website. The ones who already know this, you don't need to listen to this, because you all know about the Master Academy website now. masteronline.lk. Okay,
Sir,
so
in the
yes,
booklet it says the monthly fee is 1,800.
1,800. Yeah, then you can pay half of it. That is for grade 9, I think they have put 1,800. I'm not sure what amount they have put exactly. You can check and see. If it's 1,008, then you can pay 900 rupee, Puta. Okay, this month you all can pay 900 and come. So once you, this is the sign up thing. This is first time users. You can put all the details and you can sign up. After that, you will get registered. After that only you can purchase it. I'll just show you. Once you go to the classes, you have local syllabus, Edexcel and Cambridge. Go to local. You all are grade 10 now. Select the grade 10 class, not the papers. Paper later only we will be starting it. Here grade 10 papers are there. That is later. For now, this is the grade 10 theory class. Click on it. Even through phone, you all can do it. Then can you see all the subjects? Maths is there, commerce is there, ICT, science, Sinhala. Maths is done by Rizlan sir. He is the most famous maths English medium sir. You all know from Colombo. And commerce is done by Faizan Jeffrey sir, one of the financial analyst, superb ICT, this commerce teacher. And Sir Yusuf Riaz is doing the ICT. He is the one who made our website also. And science is done by me. Sinhala is done by two teachers, Ramesh sir and Madhubhashini miss. You all can check their classes and you all can go to them, whoever you need. It's all online. Physical also we have at Kolonnava and Vellavatta. So Colombo students can join the Vellavatta classes if you are close to Vellavatta, the Hivala area. Kolonnava classes if you are close to Dematagora, Maradana, Kolonnava area, you can join to that institute. Out of that, outstation students or some students who can't come to the physical classes can join online. So when you click on science, it's not showing because I have not registered yet. Okay, like a student, I have to register and you can purchase from here onwards.
Excuse me, sir. Do you all
Yes,
have
Puta.
Arabic literature?
Arabic literature, we don't have a teacher.
Okay, sir.
Yeah, I
Then
think in the
Mazahira
website
teacher.
it shows. Yeah, Mazahira teacher, I think she still didn't start the classes. You can ask from the institute this. There is a whenever you get stuck in the website, there is a number to be contacted. You can ask them, or I'll just later ask and tell you all. Right?
Sir,
Okay,
at what
so
does the Vellavatta science class start?
Vellavatta is on Sundays. Vellavatta Sunday, 8:30 to 11. Science classes. Then 11 to 1:30 maths classes will be there. So if you come on Sunday, math science both you can cover on the same day. This is for grade 10 Vellavatta students. Hari. Then Puta, that part is done now. If you all get stuck now, within this week itself, you all can purchase the classes. If you all get stuck, new students, especially new students, if you all get stuck, make sure you contact me. I will try my best to guide you all in the website and get the needed things. Grade 11 students who are coming to grade 10 classes can pay half the fee amount. Half the fee. Okay.
Sir, for the physical also, is it the same price?
Yeah, physical, the fees is little bit different. Grade 11, 2,400. Grade 10, 2,200. Grade 9, I think it's 2,000. Grade, for the physical class students also, grade 11 students who are coming to grade 10, we normally charge half the fee. Especially for science, it's always half the fee. So anyone who wants to join physical, you all can join, no problem.
Sir, for
Yes,
online, do we have to
today we'll
purchase
start
the
a
ticket?
little bit at least. No, once you purchase classes, we will send the tute freely.
Sir, do we have to pay for the
Puta, this payment thing, you all can send me a WhatsApp message because, yeah, if you have any personal doubts related to payment thing, you can send me a WhatsApp message. Normally, it's like this. The grade 10 students directly you all can purchase. Grade 11 students, you all can purchase the grade 11 full by paying the full fee, and while purchasing grade 10 you can pay half fee. That you have to do a bank transfer, bank deposit, because if you are trying to pay from card, they will think it's your like full fee. You all a grade 10 student killer, it might take the full fee, so try to pay from the bank deposit method or something. Grade 10s, pure grade 10s, no problem, any method you can do the payment and you can purchase the classes. If any problem related to that, you can contact me or the admin. They will help you with the if you are stuck with the payment, or if you are unable to enter to the website or something like that, they will help you. Let's come to our second most important organic compound. One big organic compound done, carbohydrate. Now protein. You all know when boys go to the gym, what do they tell to eat? If you want to grow the muscles are protein. So we have to eat protein, eggs or protein rich food, because our muscles are made out of protein. So if you look at the essential, if you look at the body components, protein is an essential component, essential part of the body of living organisms. 17% of the human body is composed of protein, because a large amount of protein is there in different places. We will study one by one where the places is. In different places, different types of proteins are available. Okay. The elemental composition of protein is carbon, hydrogen, oxygen, nitrogen. The ones who have the tube, page number 9, put these things: carbon, hydrogen, oxygen, nitrogen. Those are the elemental composition of protein. Always these four should be there in protein. Compulsory. Sometimes sulfur will be there, but sulfur is not compulsory. So what to write in last part of page 8? Last part of page 8, last week itself we completed. If you watch the recording, you will be able to get it. That is related to photosynthesis and cellular respiration. Already we have discussed the cellular respiration today. The ones who are not there for the last week's class, please watch the recording. I will upload it again in the group. Okay, once I complete adding everyone, I will again upload. Please watch it. It's very important. Then structural, complex structure made up of polymerization of amino acids. Another name for amino acid is polypeptide chain. What is the meaning of this? Wait, I will explain. Amino acid is the small component. This small component will join like this and form big component. This big component is only known as protein. So what is the building block of protein? The small components. The small component is amino acid. Are you all clear? The process of forming this is known as polymerization. Making the protein or making big compounds by using small particles is known as polymerization. Are you all clear about it? Then there is a small space
Sir, can you
in page number
explain that
9.
again?
Amino acids joining together to form a big structure. It's like small beads are there, beads. You're putting a thread and connecting all the beads and making a chain. That is the protein. If the beads are alone, they are known as amino acid. that process name is only polymerization. It's a name of the process. Okay, then now we have to see what is inside this amino acid. This one bead is there, no, one piece. Inside that piece, what are the components? Actually, inside that small piece, there are 5 components. I'll draw it here. So in the tute, I have given a box like this. Inside that box, you have to draw. This is the carbon, alpha carbon. Alpha carbon. C. It has 4 bonds. 1, 2, 3, 4. Each bond has some chemical component. This is known as amino group or amine group. Amino group. What is the amino group part? NH2. Next one, COOH. That is given a name carboxyl group. Carboxyl group. COOH. Then there is another part, hydrogen only. Hydrogen atom. Symbol is H. This last part, last bond, we call it variable. R. Symbol is R. Variable. This variable may be any component. Now this is fixed. Always one hydrogen, one COOH, one NH2. This might change. So that's why they have kept it as a variable. This whole thing is only one amino acid. Can you all quickly draw the structure? Later I will teach you all what are the things which can come under this variable. Don't worry, I will teach that part. This is the structure of amino acid. Once you all are done, tell me. These are names. Don't ask me what is carboxyl group. Those are names. Carboxyl group has 1 carbon, 2 oxygen and 1 hydrogen. Those are names. NH2 is known as amine or amino group. If you are done with this, we'll go to the next part. Okay, this variable R group represents a group containing carbon, hydrogen, or both carbon and hydrogen. Depending on who comes here, there are 20 different people can come here. For example, there can be a hydrogen. Actually, that is known as glycine. Like that, glycine, myrcene, thiosin, Like that 20 amino acids are there. But good time for your O level, you don't need to memorize all 20. A level, you have to memorize all 20. O level, you need to know only one fellow, the simplest amino acid. Okay. In the simplest amino acid, instead of this R, we put one hydrogen. That is the simplest amino acid. In page number 9, below these diagrams, I have given the protein picture. Now you all wrote this, no? Alpha carbon is C, amine group is NH2, carboxyl group is COOH, hydrogen atom is H, variable group we give R. Later, this can be a hydrogen, this can be a carbon, this can be carbon hydrogen. Like that, it can be any 20 things. But you don't need to memorize all 20. You need to know only the simplest fellow, that is hydrogen. There can be 20 different amino acids due to the change in this group. Therefore, all the protein present in the whole world, maybe bacteria's protein, plant protein, animal protein, are made up of the mixture of these 20 amino acids. They mix in different patterns and form protein. The simplest amino acid is glycine. Here you can see the structure of glycine. Okay, I will draw the proper one. See, carboxyl group is compulsory. Amine group is compulsory. Hydrogen group is compulsory, no? This is only variable. Here, if hydrogen is there, if hydrogen is here, then his name will be glycine. This you have to remember. Only this you have to remember, glycine. Like that, there are another 19 more names. Good time for our O level. It's not there. You don't need to memorize it. Variable means changeable. In nature, there are different varieties. These are like permanent. They won't change. But this part, it may be changeable. That's why they call it variable. And they are giving you one example of it, glycine. You have to draw that in page number 9. Will the extra knowledge part come for the exam? Yes, some of the extra knowledge part directly they are asking. Okay. Then this non-essential amino acids and essential amino acids. Some students think non-essential means not needed, essential means needed. No, no, no. Whether it is essential or non-essential, both are needed by the human body. But the difference between non-essential amino acid and essential amino acid is this non-essential amino acid that can be produced in our body. So that means whatever protein you eat, these non-essential fellows can be somehow produced. But essential amino acid, they can't be produced in our body. Therefore, directly by eating the food, we have to get the essential amino acid. That's why sometimes, vegetarians, they don't eat beef, no? They don't eat beef, they don't eat chicken, they don't eat fish. So, some of the essential amino acid is there in meat, fish, chicken. So, their body can't produce it. They don't eat it directly through food. So, sometimes they become very weak. Sometimes they get a certain type of disease. So therefore, scientists have made a tablet with the essential amino acids for the vegetarian people. You all don't need to know all those things. I'm just telling you, you all should know that essential amino acid, non-essential amino acid, both are needed for our body. Essential amino acid, directly you have to eat it from the diet. Non-essential amino acids, no problem. Whatever protein you eat, your body will produce the non-essential part. But essential part, you have to take it in your diet. Are you clear? So both are needed for our body. One directly from food you have to take, other one automatically your body can produce it. So I think with that, yeah, we have to fill another one more chart before formation of polypeptide chain. Already you all know this. Before that, shall we fill this chart? Protein present in the muscle. Already I have given you all the answers, no? Actin and myosin. Page number 10. Write it down. Actin and myosin are the proteins which are present in the muscles of animals. Osin is the name of the protein present in the bones. You should know all those things. Hemoglobin is the protein present in red blood cells. Keratin is present in hair, nail, horn, and also feathers of birds. Legumin is a protein present in leguminous food. Can you tell me an example of leguminous food? Yes, beans. Bonchi. Those are leguminous foods. Gluten is the name of the protein present in wheat. Wheat flour, those kind of things have one protein, gluten. Albumin is the protein which is present in egg white. Casein is a protein present in milk. And there is another one that I have not given in this slide, I have given in the tube. Protein present in connective tissue. And also in the skin. What is that known as? Collagen protein. Can you all write these names? Actin myosin in muscles. Osin in bones. Hemoglobin in red blood cells. Keratin in hair, nail, horns, feathers. Legumin in beans, legume plants. Grade 9, can you all remember what is the special bacteria living in leguminous plants? In the root nodules of leguminous plants, one bacteria is living, no? Grade 9 first lesson you would have studied. Rhizobium bacteria, good. Next one, protein present in wheat is gluten. Protein present in albumin is egg white. Protein present in milk is casein. Protein present in connective tissue and skin is collagen protein. 1, 2, 3, 4, 5, 6, 7, 8, and 9. 9 places, 10 proteins. Not because actin, myosin, 2 proteins are there. These you all should know. Even though they are given under extra knowledge in your textbook, in some places in your past papers, provincial papers, they have asked questions related to it. Yes, with this chart, we can end the session. Next week, we will start with identification of protein. For that, we have only one test, Buret test. So again, we are meeting on Monday, 7:30 p.m. That will be the third week. So you all can purchase the classes from the website and join the session. Yes, any doubts to be asked? If you have any doubts, please ask. Okay then students, inshallah, we are hoping again we will be meeting on next Monday. So make sure you purchase the classes from the website early so that last moment these inconveniences won't be there. If you have any problems related to the website when you are joining or anything, you just send me a screenshot to my WhatsApp. I will guide you all how to proceed. Yes, when will the paper class happen? Paper classes, don't worry, soon after the Ramazan festival, we will start the paper classes. Is there any registration fee for the website? No, no. Online, there is no registration fees. Directly, you can register freely, but when purchasing the class, you might have to do the payment. So should we memorize the structure of glucose molecule? That part you don't need. The structure of the glucose molecule formula you need it. They have given a structure like this. That part you don't need. But formula C6H12O6, yes, compulsory, that is needed. Any type of monosaccharide, this is the same formula. Glucose, fructose, galactose, same formula. Maltose, lactose, sucrose will have what formula? C12, H22, O11. Yes, any other things to be clarified before we wind up the session? Are you all clear about the session? Because it's a very long lesson, more new, all are new, no? So your workload is more. You have to go, like you have to again and again revise and get familiar with the names as soon as possible. I will also try to send the tudes so that it will be more easy. Next week when you are coming, you will have the tude, you would have watched the recording. You all are coming purely to learn the real essence of science. Okay then, Assalamu alaikum and a very good night to all of you. You all do a small revision and then you all can have dinner and have a good night's sleep. Don't forget to do a slight revision before sleeping. And one thing, please inform your friends about the classes. Okay, so that they will also get benefited from the sessions. Okay, then meet you next week.